Welcome to Chapter 14 of Microbiology. So this chapter is introducing you to the host immune defenses and will lead greatly into Chapter 15, which is our adaptive immune defenses. The system of immunology is the study of our immune response to foreign pathogens. And so as a consequence, our immune function consists of surveillance of the body, recognition of items that are foreign, and tolerance to items that are self, and destruction of foreign entities. And so you can see here the interaction between our white blood cells and our self cells. There's absolutely no reaction. However, here it's shown that this is indeed a foreign interaction between the white blood cell and a pathogen recognition receptor. This recognition of the pathogen-associated molecular patterns on the microbe initiate the phagocytosis and destruction of that foreign object. The host defenses are broken into innate or adaptive. So our innate immune defenses are those that are present at birth. They are more nonspecific. So they are specific for foreign pathogens, but they are less specific than that of the adaptive. And what I mean by that, the innate immune responses, if you see a microbe, you will respond the same the first time as you will the second time or the fifth time. Whereas with the adaptive immunity will actually change and become more specific to the distinct microbe at each exposure. Therefore, the adaptive immunity is how we can actually have long-lived protection. An excellent example of this is of chickenpox. If you've had chickenpox as a child, you developed an adaptive immune response during that time, and now you are protected against chickenpox. We now vaccinate against chickenpox, and so instead of having to have the infection, we obtain adaptive immunity through vaccination. You can see here in this chart that our first line of immune defense and our second line of immune defense are in the innate category, so all in Chapter 14, and they're listed here. The third line of immune defense is that of the T cells, B cells, and they're highly specific, and those will be discussed in Chapter 15.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
So this video covers the entire sections of the immune system. So this is Chapter 14 and 15, and so it's everything that you'll learn over these two lectures. So the immune mechanisms of the host, we break those down into first, second, and third lines of immune defense. Our first line of immune defense are things like our skin, which act as a nonspecific barrier to all microbes that would enter into our body. Second lines of immune defense consist of phagocytes, inflammation, inflammatory mediators that alert the body that we have a foreign invader. Again, these are still somewhat nonspecific. The third line of immune defense is that of our B and T cells, and as a consequence, we develop memory due to these adaptive cells. They're highly specific, and they're the reason that we can vaccinate and be protected against diseases. Physical barriers fall within the first line of immune defense. The example we were discussing before is the skin. You can see here that the skin has sweat glands, has a low pH, and so as a consequence of this, the microbes that will inhabit the skin are distinct to the skin. And so as long as the skin remains intact, you are free from infection. Additionally, we have things such as our tears, saliva, and urine, which actually physically flush out microbes. So if we happen to get something in our eye, our tears will flush away this item along with our eyelashes. Urinary system, because the buildup of fluid in our bladder then will rapidly push out fluid, right, relieving the body and maintaining homeostasis. But this also prevents the colonization of microbes in the urethra. We have lots of goblet cells that produce mucus that line the respiratory tract, and we have cilia that then move that mucus back and forth to protect the area of the airways. In addition to these other first lines of immune defense, we have our resident flora. So our resident flora is going to take up space, preventing the colonization by microbes that could be pathogenic. A physical barrier in our respiratory tract is that of both the mucus as well as our nasal hairs, which trap large particles, and the cilia that will then brush the mucus along, keeping the particles out of the airway. The chemical defenses of the first line of immune defense consist of our sebaceous glands, so all of our oil secretions, lysozyme present in our tears. It can actually hydrolyze the cell wall of bacteria. Lactic acid and electrolyte concentrations in our sweat will actually inhibit the growth of certain microbes. The skin has an acidic pH, so things such as Staphylococcus epidermis are specifically qualified to grow in this acidic pH, but then things such as fecal bacteria are inhibited. Hydrochloric acid in our stomach, so any microbe that needs to colonize the gastrointestinal tract, such as acid acidophilus, must be able to survive the hydrochloric acid in the stomach, moving through to get to the gastrointestinal areas. Digestive juices by bile, so the intestines are protected by the bile secreted by the gallbladder. So any microbes that are able to colonize the intestines must be able to survive the bile. Likewise, the vaginal cavity has an acidic pH, preventing the colonization of microbes that cannot tolerate this pH of 2 environment. So you can see here the areas where malaria is most prevalent very similarly matches that of the areas where people have a higher incidence of sickle cell anemia. Additionally, pathogens tend to have a high specificity for their host cells, and so malaria being infective of blood cells, Plasmodium vivax distinctly interacts with the Duffy blood group antigen on our red blood cells. So people who lack this Duffy blood group antigen cannot be infected by malaria. And so therefore you see that similar types of incidences would occur. Which of the following is not an example of a first line of defense? White blood cells, lysozyme, hydrochloric acid in the stomach, the sneeze reflex, or cilia in the respiratory tract? The answer is white blood cells. The immune system has quite a lot of terminology. For example, our white blood cells include leukocytes that are broken into granulocytes or agranulocytes. For example, our granulocytes are things such as neutrophils, eosinophils, and basophils. All of these cells have granules in their cytoplasm, hence they are termed granulocytes. Monocytes and lymphocytes, however, have no granules in their cytoplasm. Therefore, they're termed agranulocytes. Our immune cells have to be able to recognize non-self from self. Non-self is indeed foreign proteins or foreign materials that are located on the surface of microbes. Self proteins, however, are commonly produced and have to be recognized by our immune cells, and no response must be elicited. When we think about the non-self proteins, there are things such as pathogen-associated molecular patterns. Pathogen-associated molecular patterns are on the surface of microorganisms and are used to distinguish these microorganisms from self-proteins. Pathogen-associated molecular patterns, or PAMPs for short, are recognized by pattern recognition receptors. These are on the surface of our white blood cells, and so these pattern recognition receptors will bind to the surface of the PAMP, and as a consequence of that, recognition will take place. Our blood is a primary component of our lymphatic system. So the blood is broken down into predominantly plasma. Then there's a small amount of cells and formed elements. So when we break this down into proteins, the proteins that are circulating in our blood include albumins, globulins, and fibrinogen. Albumins are going to be necessary for regulating viscosity and osmolarity of the blood. Globulins are our antibodies. So these are really important for long-lasting immunity and will be discussed further in Chapter 15. Fibrinogen is a clotting factor. So fibrinogen is necessary for breaking up the area and clotting so that the pathogen cannot move to a new site of infection. Then, of course, the other solutes that are located within our blood include various ions such as sodium, potassium, and chloride, which maintain osmotic balance and pH, as well as gases such as oxygen and carbon dioxide that are necessary for normal metabolic processes. Our white cells that break up the blood consist of predominantly neutrophils, a small amount of lymphocytes, and then fewer monocytes, eosinophils, and basophils. The innate immunity is consistent of the neutrophils, monocytes, eosinophil, and basophils. This is all Chapter 14, and we'll be discussing these further. The adaptive immunity is that of lymphocytes, and these are in Chapter 15. Hematopoiesis is how all of our cells arise, and we begin with a bone marrow-derived hematopoietic stem cell. Our hematopoietic stem cells can differentiate into either common lymphoid progenitors or common myeloid progenitors. Common lymphoid progenitors are T cells and B cells, as well as our distinct plasma cells necessary for secreting antibodies. These are going to be discussed further with our adaptive immunity. Common myeloid progenitors consist of our erythrocytes, so red blood cells necessary for carrying oxygen, our megakaryocytes or platelets, which are necessary for other metabolic functions, and then of course our various forms of innate immunity, so basophils, mast cells, eosinophils, neutrophils, and then of course a whole host of monocytes, macrophages, Langerhans cells, and dendritic cells. These all consist of antigen-presenting cells. Hematopoiesis consists of the production of all of our blood cells, and so this takes place in the bone marrow, and what we find is the hematopoietic stem cells are multipotent, meaning that they can become any blood cell in the body. Embryonic stem cells, in contrast, however, are totipotent. These cells can become any cell in the body, including neurons, lung, cornea, liver, and kidney. So they can become entire organs. So the ability of stem cells, or specifically embryonic stem cells, they have a much larger capability of differentiation. Hematopoietic stem cells, located in the bone marrow, however, only have a multipotent or a smaller quantity of differentiation capability. So our leukocytes differentiate from bone marrow, and distinctly these are considered either granulocytes or agranulocytes, as discussed before. Granulocytes, such as neutrophils, are the most predominant immune cell within our body. These cells are highly phagocytic. They are indeed the first line of immune defense, and they initiate the inflammatory cascade, letting us know that we have encountered foreign material. I correlate neutrophils to things such as pawns in a chessboard. Eosinophils, however, are very, very rare. So these are distinctive pieces such as a queen or a king, so something that you have only an N of 1. So eosinophils are necessary for destroying eukaryotic pathogens. Can you think back to a eukaryotic pathogen that we discussed before? Examples include tapeworms, roundworms. IgE is indeed an antibody response. So IgE is part of our adaptive immunity, but IgE works very closely with our eosinophils to mediate the degranulation and allergy response of eosinophils. Basophils, these guys are highly rare. So these are the rarest immune cell that we have within our immune system. They do also participate in allergy responses and facilitate degranulation using IgE, an antibody. Mast cells are non-motile cells, so these guys are located in tissues and they're tissue bound. They participate in allergy responses, so they're going to be located in sites of allergy response such as the skin, the respiratory tract, or the nasal cavity. You can see here just a brief overview of the allergy response. We'll discuss this a bit further when we get to chapter 15. Agranulocytes, or cells lacking granules that are part of the innate immune system, consist of monocytes, macrophages, and dendritic cells, of which we'll discuss further. Monocytes and macrophages and dendritic cells are all the largest white blood cells that exist in the immune response. They tend to have a kidney-shaped nucleus and they're highly phagocytic. Monocytes are indeed the earliest stage and are precursor cells to both macrophages and dendritic cells. So in the comparison, if you were to rank them, these are the least phagocytic cells, and as a consequence of being able to phagocytose a bacteria or infectious agent, they are then going to be able to only digest and present that infectious agent on their cell surface at a lower level. I tend to compare this to if you were to eat a large quantity of food and you were to get sick, you're going to vomit and push more stuff out of your body because of the quantity of food you digest. So monocytes eat less. As a consequence, monocytes then produce less receptors on their cell surface for presentation to other cells. Macrophages, in the same line, they're more phagocytic than that of monocytes, right? So they're intermediate, and then they are less phagocytic than that of dendritic cells. So these guys are going to be able to be the final differentiation of a monocyte, and they're going to be able to present antigens to T cells. So you can see the macrophage that is differentiated, and you can see that there's 5 receptors that it's using to activate this T cell. Dendritic cells, however, are the professional antigen presenting cell. And so these guys have 8. They have 8 receptors that they're using to activate this T cell. These receptors are going to elicit a much larger response from the T cell than that of those receptors possessed by this macrophage. So as a consequence, the T cell will become activated faster and elicit a more robust immune response than that of the T cell stimulated by the macrophage. So the agranulocytes that consist of the adaptive immunity, our lymphocytes make up about 20 to 35% of our immune response. Our lymphocytes can be either innate or adaptive. Examples of innate immune T cells are those of the natural killer T cells. So natural killer T cells are able to kill infected cells within the innate immune response. The difference is they maintain the same specificity throughout the infection, so regardless of seeing the microbe 1 time or 5 times, these natural killer cells will respond the same. The adaptive immunity, however, differs in the sense that you have humoral or cell-mediated responses. Humoral responses are that of our B cells, memory B cells, and plasma B cells. So memory B cells are B cells that have been activated by T cells, shown here, and as a consequence, they can be activated and programmed and saved for a long-lasting immune response for later. Plasma B cells, however, are B cells that have been activated by a T cell, but then they're now going to move into a plasma differentiated state and secrete antibodies. Cell-mediated immunity, such as T cells. So our T cells are differentiated into various T cell types. Cytolytic T cells kill foreign cells. These are typically CD8 killer T cells. So CD8 cytolytic T cells. We also have a variety of helper T cells, and within the helper T cell family consists of standard helper T cells, which will modulate and activate the responses of other cells, such as these helper T cells activating this B cell, or we have T regulatory cells. So T regulatory cells are a class of helper T cell that will turn off immune responses. So in this case, if this were a T regulatory cell, it would tell this B cell, no, you don't need to respond to that microbe, and would turn off these receptors, and the B cell would go off and not secrete antibodies. The memory T cells, these are T cells that have been activated by a mature DC, or mature dendritic cell, and now these activated memory T cells will move on and be able to respond to the microbe at a later date. Here's a quick breakdown of the leukocytes. So neutrophils, eosinophils, basophils, monocytes, and lymphocytes, and the information that we have discussed on the previous slide. Feel free to review this at a later date. So erythrocytes, or our red blood cells, they do indeed develop in the bone marrow from bone marrow stem cells. They have no nucleus, but they're really important for carrying hemoglobin, which has a high affinity for oxygen. So the carrying of hemoglobin maintains the body's homeostasis. Platelets, however, these guys are necessary for clotting. And so in particular, when we think about the combination of platelets and fibrinogen, they're necessary to wall off microbes so that they can't disseminate that infection to other sites of the body. Our lymphatic system. Our lymphatic system actually provides an axillary return for that of the extracellular fluid from the circulatory system. So when our circulatory system reaches a high osmotic threshold, it can actually drain off some of that threshold into the lymphatic system, as shown here. Additionally, the lymphatic system allows for immune surveillance because it runs in parallel with that of the circulatory system. And as picking up the drain off from the circulatory system, it can pick up foreign materials that may be trafficked by immune cells. So our lymphatic system, as it permeates through the body, you can see that it is available for a quick pickup of extracellular fluid that may have been drained off from the cardiovascular system. The flow is one directional, and eventually even the lymphatic system running in parallel runs flow one direction toward the heart, eventually returning blood to the bloodstream. Classification of the lymphatic organs. We break the lymphatic organs into either primary or secondary lymphoid organs. The way that you can distinguish this is that primary lymphoid organs are always present. Primary lymphoid organs consist of our bone marrow, the site of hematopoiesis, or our thymus gland. This is the site of T cell development. Our secondary lymphoid organs are that of a site that is necessary to educate our lymphocytes and other immune cells due to the invaded microbe. The lymph node and the spleen are consisting of secondary lymphoid organs. That's because these lymphoid organs actually swell and change during infection. So you'll note that if you have an upper respiratory infection, the lymph nodes around your throat will swell. Individuals who suffer from things like Epstein-Barr virus, the causative agent of mononucleosis, may have splenomegaly and have issues with their spleen because all of the immune cells are going to the spleen for antigen presentation. Primary lymphoid organs, such as the bone marrow, is the site of all blood cell production. For example, there's more red bone marrow in children, and this bone marrow will actually decrease as we age. In the bone marrow, some white blood cells actually mature here, such as our B cells. The thymus gland, however, the thymus gland is where our T cells are going to mature. It is indeed largest in children and shrinks as we age. Immature T cells, after they have moved from the bone marrow, will actually enter the thymus gland and will mature there. My example for this is often when you think of students who move from the high school level, right? They're still pretty immature, and then they enter college. So once you've entered college, you're going through a maturation process. So this maturation process should allow you to obtain a career. And so you're likely trying to become some sort of clinician. So at this stage, you're going through the maturation process with your education. So T cells mature in the thymus, and they become distinct types of T cells with distinct careers and functions. Our secondary lymphoid organs are our lymph nodes, spleen, and gut-associated lymphoid tissue. Our lymph nodes, as we had discussed before, do actually swell during infection, and they swell because this is a site of antigen presentation. Antigen presentation is where immune cells are showing T cells the type of infection that they need to respond to. And so as a consequence of this, our immune cells will congregate within this conference type center, the lymph nodes, for antigen presentation. The spleen, likewise, is also a site of antigen presentation. It takes place more so with B cells, and so in this scenario, you have immune cells that will congregate in the spleen and perform antigen presentation to tell these cells within that area what type of infection is present. Gut-associated lymphoid tissue, or GALT. Our gut-associated lymphoid tissue, if we consider all of the foods that we eat on a day-to-day basis, it is not surprising that we would have lymphoid tissue, such as gut-associated lymphoid tissue or Peyer's patches, in our intestines. And what you see here is this forms a structure very similar to what you would find in a cross-section of spleen or lymph nodes. This gut-associated lymphoid tissue is necessary for processing all of the food antigens, so all of the food proteins that are broken down, showing those to immune cells to make sure that that food was indeed safe. T cells mature in the lymph nodes, spleen, thymus, gut-associated lymphoid tissue. Thymus. So the second line of immune defense has a series of information. And so the second line of immune defense consists of recognition of foreign particles by our innate immune cells, initiation of inflammation, phagocytosis of foreign molecules, interferons or alerting proteins that specifically tell us what type of immune response needs to be initiated, and complement proteins, which will both increase phagocytosis and the lysis of foreign membranes. So those pattern recognition receptors that we were discussing before are literally that alert system. They let us know that foreign microbes are present, and they let us know that we need to initiate an immune response. And so the pathogen recognition receptors are on the surface of our host cells, right, shown here. This is a macrophage expressing a pattern recognition receptor, and a specific one in particular called a toll-like receptor. Toll-like receptors, especially toll-like receptor 4, is specific for recognizing lipopolysaccharide on the surface of gram-negative bacteria. And so what you find is that lipopolysaccharide on the surface of gram-negative bacteria is generally termed a pathogen-associated molecular pattern, PAMP for short. And so pathogen-associated molecular patterns are things that exist on the surface of bacteria or viruses, but do not exist in the host. So excellent examples of this are lipopolysaccharides on the surface of gram-negative bacteria, double-stranded RNA, which does not exist in our host cells, and peptidoglycan, which is present on the outer surface of gram-positive bacteria and in a smaller amount present in gram-negative bacteria. The macrophage will use its pattern recognition receptor, shown here, to bind the pathogen-associated molecular pattern. The binding of that pattern will initiate a various set of responses. It will initiate phagocytosis of the foreign molecule, and then once the foreign molecule is broken down and digested, that foreign molecule can actually be used to do antigen presentation, which we'll discuss further. Additionally, the macrophage that digested the foreign material will then secrete inflammatory mediators, interleukins, and cytokines. So these cells possessing these PRRs will secrete cytokines and chemokines. Cytokines are alert signals that tell us what type of infection is present. And so the cytokines, an example of this is tumor necrosis factor, and it will be secreted after binding peptidoglycan on gram-positive bacteria. Chemokines, these guys are actually chemical signals. I call them chemical breadcrumbs. These chemical signals will induce the movement or trafficking of immune cells to the site of infection. That movement is called chemotaxis. So chemokines elicit chemotaxis, or the trafficking or movement of immune cells to the site of infection. An example of this is interleukin-8, which will induce the migration of neutrophils to the site of infection.
Of phagocytes, called pattern recognition receptors, make it possible for the phagocyte to immediately recognize a number of different components of infectious agents, referred to as pathogen-associated microbial patterns. Toll-like receptors are a class of pattern recognition receptors that play a critical role in the early innate immune response to invading pathogens. Several different toll-like receptors located in the cell membrane or endosomal membranes of phagocytic cells have been identified. They recognize lipoproteins and lipoteichoic acids, viral double-stranded RNA, lipopolysaccharide, and bacterial flagellin. Some toll-like receptors exist as monomers, whereas others exist as dimers. When a toll-like receptor interacts with a particular pathogen component, it activates specific cytoplasmic adapter proteins such as MYD88, MAL, TRIF, and TRAM. This activation initiates a signaling pathway to defend against a specific type of pathogen. Lipopolysaccharide is a pathogen-associated structure produced by gram-negative bacteria. It first binds to lipopolysaccharide-binding protein and then associates with a cell surface protein, CD14. The CD14 with bound lipopolysaccharide-binding protein then activates TLR4. The activated TLR4 then interacts with and activates specific cytoplasmic proteins. The interaction of TLR4 with these cytoplasmic proteins activates other proteins, allowing them to convert inactive transcriptional activators such as NF-kappa B into active forms. The activated NF-kappa B moves into the nucleus and binds to specific sites on DNA. Binding of NF-kappa B to specific sites on DNA induces transcription of genes for cytokines, including interleukins and tumor necrosis factor. These cytokines move out of the phagocyte and stimulate various aspects of both specific and nonspecific immunity.
So don't worry about the distinct mechanisms that are taking place underneath the cell. The things that you should actually focus on is that a pathogen-associated molecular pattern, or microbial pattern as the video describes, is necessary for binding to a pattern recognition receptor. In this case, the example is a toll-like receptor. And that binding actually initiates a signaling cascade that allows us to secrete cytokines and chemokines that move cells to the site of infection. The inflammatory response consists of four major classic signs: redness, warmth, swelling, and pain. Redness essentially is taking place because there's increased circulation at the site of infection. So this example shown here shows that there were pathogens on the surface of this nasty splinter, and those pathogens, upon being recognized by macrophages, those macrophages are going to do the process you saw in that previous video. The macrophage will bind to the pathogen-associated molecular pattern using the pattern recognition receptor, and will then secrete things such as chemical mediators, cytokines, and chemokines. Right? The cytokines will then tell other immune cells what type of infection is present, and so then they will respond appropriately. And then these macrophages are also secreting chemokines. The chemokines are moving down here toward the vasculature, and they're going to be responsible for step 2, shown here. So the increased circulation and vasodilation allow then the immune cells to come to the site of infection. because of the increased blood flow, warmth takes place. Swelling, if you have all of these immune cells moving to the site of infection because they're trying to get rid of this bacteria, you find that you have to swell within that site of infection because of increased fluid and increased migration of cells. And then, of course, we have pain. Pain takes place because of the stimulation of the nerve endings within this breached section of skin. And so the secretion of chemokines allows the diapedesis, or the movement of these cells out of the vasculature into the site of infection. And so the chemokines are necessary to properly kill this invading bacteria. And so you can see finally the wound has closed. The bacteria are being phagocytosed by monocytes, macrophages, or dendritic cells. And then the movement of macrophages, monocytes, and dendritic cells into that site of infection is much fewer. So the chemical mediators of an immune response, of course, have vasoactive functions, have initiating events, chemotactic action, so movement into the site of infection. It's highly orchestrated, so this is just to show you the overall coordination of efforts to elicit an immune response. Please do not try to memorize this slide. I had mentioned diapedesis in the inflammatory slide. So you can see here there are neutrophils that happen to go through the vesicles, and because of the chemotactic factors, or the chemokines that were secreted at the site of infection, these immune cells are now going to slow down. They're going to find junctions between cells, and they're going to squeeze through. Chemotaxis is the migration in response to chemokines. Again, I call these chemical breadcrumbs. They're going to recruit immune cells to the site of injury or infection, and they're going to act as a gradient. So as you move closer to the site of infection, you're going to find higher concentrations of chemokines being secreted by immune cells. Fever. Fever can both be beneficial and detrimental to the host cell body. Circulating pyrogens can reset the hypothalamus to increase the body's temperature. These circulating pyrogens can come from two different sources. For example, they could be exogenous pyrogens, meaning that they are obtained from the foreign agents such as a prokaryote or a bacteria, or they could be endogenous pyrogens. These are pyrogens that happen to be liberated by our immune cells, such as monocytes, neutrophils, and macrophages. Examples include interleukin-1 and tumor necrosis factor. So you can see here this macrophage has secreted our awesome pyrogens trying to fight the infection. The induction of fever can inhibit the multiplication of temperature-sensitive microbes at the site of infection. They can also impact the nutrition obtained by bacteria, so they reduce the available iron. Additionally, increased temperatures increases our metabolism, and so this stimulates immune reactions and protective physiological functions to take place at a higher frequency. Phagocytosis must take place for antigen presentation to occur, and so our phagocytes, neutrophils, monocytes, macrophages, dendritic cells, these guys are necessary to get rid of invading materials. So for example, these phagocytes will move to the site of infection due to chemotaxis, responding to chemokines. Once they are at that site of infection, they will bind the pathogen-associated molecular patterns with their pattern recognition receptors on the surface of their host cell. And so once they bind that bacteria, they can engulf the bacteria into a vacuole. That vacuole then is called a phagosome. The phagosome merges into the phagolysosome, so it is the phagocytic vacuole moving and merging with the lysosome. And then as a consequence of that, those components can be digested and broken down, and so we can release those debris, or we can actually take those microbial components and we can use them in antigen presentation. So we extract the immunogenic information from those digested bacteria, and we do that so that we can show other immune cells what type of bacteria we need to fight for later.
Attracted to the area of invasion by chemical products of the microorganism, phospholipids released by injured mammalian cells, or by components of the complement system. The phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibody, lipopolysaccharide, and complement receptors. For example, the C3b component of complement coats bacteria or other particles and then binds to C3b receptors on the phagocyte. This process of coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuole known as a phagosome. Vesicles in the cytoplasm called lysosomes fuse with the phagosome, releasing digestive enzymes such as lysozyme and proteases into the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed and digested. Finally, the digested contents of the phagolysosome are eliminated from the phagocyte by exocytosis.
So macrophages are really critical in removing infectious agents from our body. Particularly, they do so best for infectious agents that are extracellular, so bacteria that have colonized the surface of host cells. However, there's lots of infectious agents such as viruses that will infect the interior of our host cells. And so for viruses, we need to elicit interferons. Interferon alpha is a very, very early interferon response secreted by lymphocytes and macrophages. Interferon beta is secreted by fibroblasts and epithelial cells. And then later, interferon gamma will be secreted by very highly activated T cells. And so I'd like you to think of these as a danger signal, an antiviral danger signal. And so it's going to be in response to things like double-stranded RNA or other viral components that are recognized by the immune system. So you can see here that this virus tried to infect this cell. This infected cell then recognized the viral nucleic acids, and as a consequence of recognizing those viral nucleic acids, it was able to secrete interferons, which then would move on and activate antiviral proteins in nearby cells. This causes the nearby cell to block viral replication, degrade the virus and its nucleic acids, and then prevent the further spread of this virus through the body.
The presence of this viral material signals the cell to produce interferon. The interferon moves out of the cell and attaches to receptors on nearby cells of the same type.
It would be kind of awesome.
The cell that produces the interferon is unable to save itself. The virus replicates in this cell and then moves out to infect nearby cells. The nearby cell that already has interferon bound to its surface responds in several ways, including production of enzymes that degrade messenger RNA and prevent protein synthesis. Thus, a virus can attach and enter the cell, but completion of the viral replication cycle is prevented.
So in addition to antiviral responses, we also have rapid and robust complement responses. Complement are distinct blood proteins that work in concert to destroy both bacteria and viruses. There are three major pathways of complement. Ways differ only in the way in which they are activated. The classical pathway, shown here, is antibody responses working with complement proteins to get rid of an infectious agent. The lectin pathway is very specific for things such as mannose-binding lectin, and it works with mannan proteins that will then interact with the microbial surface. The alternative pathway is the interaction of various complement proteins directly to the pathogenic surface. All of these pathways cause the same results. At the end of this pathway, you have processes such as inflammation, opsonization, and opsonization, shown here, is the consequence of microbes being opsonized or coated with these complement proteins, and then being coated with the complement proteins increases the rates of phagocytosis. So essentially, I want you to think of these complement proteins as, if you're a meat eater, bacon on the surface of that delicious burger, right? Bacon makes everything better, so you're more likely to phagocytose or eat that hamburger because of the presence of bacon. If you're a vegetarian, possibly it's the addition of things such as delicious cheeses on your favorite margarita pizza. But regardless, all of these are going to increase the rate at which the cells are going to consume the infective agent. And so as a consequence of increasing the rates of phagocytosis, you're also going to increase the rates of antigen presentation. Antigen presentation is when you take the microbe that was phagocytosed, you break off distinct components, and you put it on the cell's surface in a receptor and show it to other immune cells. This increases both the rates of clearance by, one, phagocytosing and getting rid of the microbe from the current location, and showing other microbes to do the same. The functions of the complement system end up recruiting additional phagocytes to the site of infection. They work very closely with antibodies and other complement proteins to opsonize the microbial surface, increasing phagocytosis. Additionally, they're going to work together to initiate this structure called the membrane attack complex. The membrane attack complex will be shown in the video next, and it is a series of complement proteins that will work together to create a donut hole on the surface of bacteria or viruses that then causes those structures to burst.
Complement acts as a chemoattractant for phagocytes. C5b reacts with other complement components including C6, C7, C8, and C9 to form a membrane attack complex. This structure forms a hole in the cell membrane and causes cells to lyse.
Clearly, the complement pathway has quite a bit of components. So for the purpose of this class, I really just want you to know that the complement pathway does initiate inflammation, creates the structure called the membrane attack complex, and additionally does function to opsonize the microbial surface and induce phagocytosis, which in turn increases the rates of antigen presentation. So which of the following defenses is most likely to be active during a viral infection? Inflammation, fever, interferon, or complement? Best answer is interferon. So this is the last overview of the major host defenses. So as a quick review, the innate defenses are non-specific. When we think about the innate defenses, the first lines of defense consist of physical barriers, chemical barriers, and genetic barriers. The second line of defense is the inflammatory response, interferons, phagocytosis, and the complement pathway. The acquired immunity, however, is the third line of defense consisting of B cells, T cells, which have either antibody production or cytotoxicity, which we'll discuss further. And these are highly specific. We'll begin now by discussing Chapter 15.